Moving in a little bit more, we have the cell wall, which again, in most bacteria is not optional, but there is that genus Mycoplasma that does not make a cell wall. For the bacteria that have a cell wall, it helps to maintain their shape, and it also helps to serve as kind of a barrier against dramatic changes in the environment. So for example, when the osmolarity of the cell changes, our cells, we don't have a cell wall, so our cells can shrink dramatically or they can burst. Because bacterial cells have that rigid structure, they don't burst. So it protects them from exploding because of changes in osmolarity. It is also the site of where a lot of antibiotics work. And the reason for this is because it's such a unique feature. Like, we don't have antibiotics, only bacteria do. And the ones that have a cell wall, they need it. And if we can prevent the bacteria from being able to make their cell wall, they will die. But it won't hurt us because we don't need a cell wall. So it's very, very important. And the chemical composition can be a little bit different depending on gram-positive or gram-negative. And we'll talk about that, what's different between them, in just a couple of slides. But first, what we'll talk about is what's similar between them. So both gram-positive and gram-negative cells have a cell wall made of peptidoglycan. And that peptidoglycan has a sugar backbone, that's the glycan part, which is a disaccharide polymer. The two sugars that make up the peptidoglycan are N-acetylglucosamine and N-acetylmuramic acid. So it's not like glucose and sucrose. No, it's a little bit more of a tongue twister to say. So N-acetylglucosamine and N-acetylmuramic acid. I'm not going to in any way, shape, or form ask you or expect you to remember the structures or anything like that, but it is a dipeptide in repeating units. And that's what they're showing you here. Brown is supposed to represent N-acetylglucosamine, and pink is the N-acetylmuramic acid. The peptido part of peptidoglycan are these peptide cross-linking bridges. So you have these side chains that can come off and then attach to these cross bridges, and that's what helps to cross-link all of these different glycan bridges. Now, I told you that there are some differences between gram-positive and gram-negative cell walls. So first we'll look at gram-positive cell walls. Gram-positive cell walls contain a very thick layer of peptidoglycan. They're trying to illustrate that here in this cartoon. But they also have this compound called teichoic acids. These are negatively charged molecules found within the cell membrane, the cell wall, and they help link it to the cell membrane. So if they link it to the cell membrane, they're called lipoteichoic acids. If they're linking the various layers of peptidoglycan, they're called wallteichoic acids. So again, that presence of the teichoic acids, whether or not they're helping link to the plasma membrane or keep the various layers linked to each other, they are unique to gram-positive cells. Now, a gram-negative bacterial cell wall is actually quite different from a gram-positive. The peptidoglycan layer is very thin, which again they're trying to illustrate in this image. There are two membranes. So there's a plasma membrane, which is just the cell membrane, right? There's also an outer membrane. And the space between the two membranes is called the periplasm. Now, technically, when we're talking about the gram-negative cell wall, we are actually talking about both the outer membrane and the peptidoglycan. The outer membrane is different from the plasma membrane. The plasma membrane looks very normal. This is what we expect. The outer membrane has all of these embedded features, and the most important of which, the one that makes the gram-negative outer membrane so unique when compared to the gram-positive, is this structure called lipopolysaccharide. And you can probably guess what it's made of. Lipids and polysaccharides. Very, very, very clever. Now, what is the role of this? Well, the LPS, the lipopolysaccharide, one is very strongly negative. That is important because it can make gram-negative cells more resistant to being consumed by the immune system through phagocytosis. It also helps gram-negative bacteria avoid parts of the immune system called the complement system, which attack and try to destroy the cell membrane. And finally, that really negatively charged lipopolysaccharide can help contribute to some of the antibiotic resistance we see with gram-negative bacteria. And that's not all. Not only does it help the bacteria be more resistant to antibiotics and help them to be more resistant to the immune system, LPS also tries to kill you. So how does it do that? Well, the LPS contains phospholipids, it contains lipoproteins, the outer membrane contains phospholipid, lipid proteins, and then the LPS. So what is it about the LPS that I said it's trying to kill you? Well, it's trying to kill you through this molecule here called lipid A. So if we're looking at a cross-section of the outer membrane, the lipopolysaccharide is right here, this kind of bluish purple. Lipid A is this one embedded within the membrane, and then there's the polysaccharides out here. Lipid A is what we call an endotoxin, and we call it an endotoxin because it's inside the cell membrane. It's not outside the cell membrane. It's not a toxin that the bacterium secretes. It's actually part of the membrane itself. So it's an endotoxin. Now, this endotoxin actually works to stimulate our immune system, specifically our inflammatory response. So when people go into what's called septic shock, a lot of times that can be happening because of the presence of this endotoxin. So when these cells lyse, or as they're dividing, they can release some of these molecules of LPS, therefore release lipid A, which causes that response in people. And so that also makes it really important for you to consider, let's say you're a physician and you're prescribing antibiotics to a patient who has a gram-negative infection. Do you want to give them an antibiotic that's going to cause the cells to lyse and release all of that endotoxin? Maybe not. So it's things that you have to think about. What are the implications clinically when you're looking at these different types of cells? So as a summary, you can compare and contrast, right, the two types of cell wall organization. Gram-positive cells have a thick layer of peptidoglycan. They have those teichoic acids. They can have lipoteichoic acids that attach the peptidoglycan to the membrane. They can have wall teichoic acids that help to keep the layers of peptidoglycan attached to each other. They do not have an outer membrane, and they have a very, very, very thin periplasm between the peptidoglycan and the cell membrane. Gram-negative cell walls have a thin layer of peptidoglycan. They lack teichoic acids. They have an outer membrane, the most important component of which is LPS, and they have a very thick periplasm. There's a big space in between the plasma membrane and the outer membrane. And we talked in the previous set of slides about the gram stains. This is something that you'll be doing in the lab. So this is what all the stuff that we've just been talking about, the difference between a gram-positive and a gram-negative cell. The staining takes advantage of those principles, those biological principles we just discussed. So gram-positive cells will stain purple, gram-negative cells will stain pink or red. So let's look at what's happening specifically. So on the top, right, this is going to be your gram-positive cell because you have that thick peptidoglycan and the single membrane. This is going to be your gram-negative cell because you have the outer membrane, the thinner peptidoglycan, and then the inner membrane. So you first apply crystal violet, which will stain all of the cells. And then you add the mordant iodine. So iodine will form a complex with the crystal violet and help it get stuck within the cell wall, within that peptidoglycan. Then you add alcohol to decolorize. Now what happens when you do that is that in gram-positive cells, in those with the thick peptidoglycan, the alcohol will dehydrate the peptidoglycan, and that kind of traps that giant crystal violet iodine complex within the peptidoglycan. In gram-negative cells, because they have such a thin layer of peptidoglycan and they have that outer membrane, what happens is the alcohol actually starts to dissolve the outer membrane and make holes in the peptidoglycan. And that causes the crystal violet iodine complex to wash away. So at the end of this step, your gram-positive cells will still be purple, but your gram-negative cells should be colorless. And then you apply the counter stain, which is safranin, that will interact with the membrane of the gram-negative cells. So that's the principle of how the anatomy of the cell wall contributes to that type of staining. There are also atypical cell walls, and we've talked a little bit about those when we talked about those mycolic acids and doing an acid-fast stain. So they're organized like a gram-positive in that they have a single membrane and they have a thick-ish layer of peptidoglycan. But then they have these waxy compounds called mycolic acids that are attached to various carbohydrates and then attached kind of out there along the peptidoglycan. This makes it difficult to stain them using the classic gram stain because the stains don't penetrate. But when you use the special stain, the carbofusion, and you're doing an acid-fast stain, that type of preparation allows the dye to penetrate and stain the cells. The other type of atypical cell wall you might see in bacteria is the one I've been telling you about that doesn't even have a cell wall, mycoplasma. They don't have a cell wall. Instead, they use sterols, right, like cholesterol, right, sterols, in their membrane to provide stability and rigidity. Archaea don't have that classic cell wall. Some of them don't have a cell wall at all. Some of them have a cell wall made out of pseudomurin. So pseudomurin still has N-acetylglucosamine, but it does not have N-acetylmuramic acid. This has N-acetyl allosaminouronic acid. I'm not going to ask you to pronounce that. It just doesn't have the same carbohydrates. So it's a different dipeptide. It also uses the L form of amino acids as the peptide cross-link bridges, whereas peptidoglycan uses the D form. The other thing that I really want to point out here is something that is going to become relevant in like just a couple of minutes. This linkage between N-acetylglucosamine and the other carbohydrate in archaeal cell walls in pseudomurin is lysozyme insensitive. So that means the enzyme lysozyme cannot break it apart. the enzyme lysozyme can break apart peptidoglycan. So when we talk about the cell wall and we talk about, antibiotics and things that damage the cell wall, the other thing to think about is, do you think an antibiotic like penicillin, which damages the cell wall, would that antibiotic be effective against something like mycoplasma? Likely not. So penicillin is an antibiotic. What it does is it interferes with the peptide bond formation. So it prevents strong cross-linking. So it makes the peptidoglycan weak. And when the peptidoglycan is weak, they can't synthesize it in the correct way. They're more likely to be susceptible to osmotic pressure, and they're more likely to lyse. And so, I told you that gram-negative cells tend to be more resistant to penicillin, partly because the LPS can kind of block it, but also one of the reasons that gram-negative cells are more resistant to drugs that affect peptidoglycan is because they have the outer membrane. So they have two membranes. So that helps to hold them together just a little bit longer and makes them less susceptible to that osmotic lysis. And the idea would be that if the bacterium can outlast your antibiotics, it could reform its peptidoglycan and be just fine. So as long as it doesn't, hold on long enough to not lyse, you actually might be out of luck. So gram-negatives are pretty nasty in that way. So this would be something that we would talk about in person, and almost certainly will be a question on your exam. Okay, why doesn't penicillin harm human cells? I'm not going to answer that. You can answer that. We've already talked about it, but I want you to answer that. I want you to think about it. The other type of damage that we commonly see to bacterial cell walls is because of that lysozyme I was just telling you about. So lysozyme is found in various bodily secretions like tears and saliva, and it breaks this bond right here between N-acetylglucosamine and N-acetylmuramic acid. And so when that bond is cleaved, that breaks apart the cell wall. So therefore, archaea, which have a different cell wall, right? They don't have this N-acetylmuramic acid, or like mycoplasma, which don't have a cell wall at all, would be resistant to the lysozyme, which has evolved to protect us from bacterial infection.